So again, thank you all. I'll just quickly whip you through the agenda so you can see what's ahead of us and what to expect from the different speakers. Of course, the first step is myself, and then Jake, who needs no introduction, our chief scientist, will go through the basis of the nucleosome technology and nucleosomics. We very firmly believe that the epigenetic signals are very, very important, and probably as important as the genetic signals, where billions and billions have been spent. And we've been really pioneering that work, particularly in blood. So Jake will go through what our technology is, why it's so special, and how well we have the entire field covered. So as it becomes more and more important, we will dominate the field. Then Dr. Axel Imhoff there will also talk about one of our new projects. We've not only now just been labelling one structure on the nucleosome, but able to target all the structures through mass spectrometry and sequencing. And he can describe some of that process work that's going through now. An incredibly exciting part of the expansion we're doing now beyond the basics of the platform. But again, it's all in the intellectual property we have or are processing. Then Jason, Dr. Terrell, will go through introducing NuQ Veterinary, which again is a very, very exciting broadening of our pipeline. It turns out that there are nucleosomes in dogs as well as humans, so he'll go through the huge market potential and how we intend to get there. And then via Skype, I guess, Heather Wilson-Ribble from Texas A&M can go through the veterinary market and how we can be involved with them and really work together to launch what would be a very exciting set of products. And then I'll just discuss and sort of bring this all together and how this works with our platform timings and the path forward. And then we'll have a brief discussion if there's time, depending on how much we take up your time in these slides. We'll have enough time for a few questions chaired by Jonas. And then we'll have closing remarks from our chairman. So that's what today's looking like. And then those who want to, we can go down and watch the closing bell, and then come up here. We'll have an hour of a mixing session. If you want to ask any particular questions to any of the team, or any of the new projects, or long-standing colorectal programs, that would be a very good time to do it. So as we always talked about, we're a very dedicated team who are completely committed to revolutionising cancer diagnostics throughout the world with routine blood tests. I'll just start with this slide. I think what this really shows our huge commitment in the last couple of years to developing the platform and really delivering on the promise that we've had, and then the fantastic technology. For that, we've really had to adapt the platform to be very robust and all the things which our scientific team can go through. That facility in Belgium is 20,000 square feet. It cost us, including the fit out, the back half, his laboratory, was about 1.8 million euros. An amazingly good deal, a vast majority provided by loans and support from the Walloon region. Our chief executive of Belgium is at the back. You can ask him questions about it. We have over 30 people working there now. Some of them you can see here. It's always important to remember, if you're a real company doing real technology, you need a large team of people doing on the coalface, actually doing the work. So as you can tell, most of the team in Belgium is female, very good lab technicians, and the space we have is really cutting edge. When combined with our large robotic machines and our technology, it's really quite something to see. So we're very happy with anyone who wants to come and visit us in Belgium. It's a fantastic facility. It's about an hour from the capital. It's quite remarkable. It's really allowed us to go to the next level of developing a really robust platform. This is a Capital Markets Day, and our Chief Financial Officer is also here, so I'll just go through the brief financials. We had $13.4 million in the bank at the end of last year. We had, through strong support of our shareholders, 6.7 million of warrants exercised, left us with about 20 million as of the start of the year, minus what we've been spending. So, and we've kept the run rate amazingly stable, around 1.2, 1.3 million a month, which is a whole different level of what most companies do when they're running large clinical trials. We can do that because of our setup in Belgium, because of the huge support we have from our clinical partners, where we pay a tiny fraction of the cost for all the trials we run. And we keep things very lean, because ultimately we're all large shareholders and we want to see a good return. So we keep it very tight. So this, just to reiterate a couple of things before we turn to the actual speeches and presentations. One thing to keep in mind, this platform has been developed. Our core project is the colorectal space. We have very large trials still being collected in the area. It's taken longer than we thought. we realised there's a huge amount of work to be done. That's why we've been serious in the last few years about the new facility and rapidly expanding the team. That work's now is coming through. We've had the first assays that have come through, and they've been very, very good, extremely reliable, very good accuracy, and we're in the process now of finishing those off, and the first results we had were excellent. So beyond the core colorectal work, we're really thickening with momentum now in lung, in the veterinary scale, and with nuclear capture, and in a range of other areas, which I'll go through when we're finished in the process. So it's a lot going on for a small company. And the reason we can do all these things on a very small budget and a reasonably small team is it's a platform, an absolute true platform, meaning 90% of the work is between all the different trials, all the different cancers, the dogs, capture is common work we have to do for all of it. So it's something we have done on a very tight budget and we're continuing to do on a very tight budget. We're not looking to massively expand at all. And just as a quick reminder, if you do find cancer early and have it removed, your outcomes are very, very good. We believe absolutely the only way of getting cancer really, truly beaten is to have 80 or 90% of people with a good screening test that can find cancer early, with a tiny blood as a routine test. And as far as we're aware, we're the only company that really has something that can be truly routine, truly done at a low cost. There's a lot of expensive, complicated tests out there in development. None of them will be truly routine in the way ours expect to be when they're products. So this is something very different. We're swimming in our own lane, and I think we can add a huge amount of value to the company, but also to humanity through better tests. So that's what we're developing. And I'll turn over to Dr. Micallef, Jake, who can start the process with talking through the technology itself, the base technology. Thank you.
Thanks, Cameron. So, as Cameron was saying, in order to make tests really routine and increase compliance so that everybody that should have a test in a screening age, for example, does actually get a test, the test really needs to be accessible to people, low cost, and not require them necessarily to go to hospital for a day just for that test. And if you look at most diseases, in order to diagnose them and monitor them, it is blood tests that are used. So, in diabetes and cardiovascular disease, kidney function, anything, most areas of medicine that you can think of, the first thing that you do is a blood test. And the reason is because it's easy, it's low cost, and doesn't involve you going to hospital for a day. On the other hand, if you look at what happens in cancer, the tests are, with the possible exception of PSA being used off-label, as it were, the tests are not blood tests. So if you want to have a look for a lung cancer, it's an X-ray or a low-dose CT scan. There's mammography scans, various other scans, or you can have an endoscopy process where somebody puts a long tube inside you with a camera on the end, or you can have cervical smears. So all of these things are very invasive and not low cost, and have compliance issues because they're unpleasant in some way. So our mission is to change that by making tests that can be run routine. So they would be, for example, like a PSA test, like a cholesterol test, where you can simply take a sample of blood and run it as a test for cancer. And the important aspects of the test for that are really that it requires a small amount of blood and that it's low cost and can be done in large numbers, which Nu-Q can be, and that's what we're doing. So what's the basis of, it's a big claim to make, so what's the basis on which we can do that? The basis is that cancer is really a disease of the nucleus. It's a disease of the DNA and the chromosomes. It's when the chromosomes go wrong and cells start to divide repeatedly and out of control that you get a cancer. This happens because of misfunction in the nucleus, in the DNA and in the chromosomes. And if you're going to look for a test, really that's where you need to look. So PSA, if you like, is the exception that proves the rule. It's a protein that's specific to prostate, which shouldn't be in the blood. And if it is in the blood, there's something wrong with your prostate. But in general, you're not going to diagnose cancer like that. You need to find that malfunction in the nucleus. And that's essentially what we do. Because when cancer cells die, these the chromosomes are digested into the individual little balls in this picture. We've got a model of one here that Professor Axel made for us. And those individual nucleosomes originating from the cancer are in the circulation in the blood. And for quite a few years now, people have been looking at the DNA that goes around them, that wraps around the nucleosome, to see if they can find mutations in the DNA that relate to particular cancers and sequence mutations related to cancer. The problem with doing that is that there are about 20 million nucleosomes in the human genome, and that sequence will only occur once. So it really is, it's a 1 in a 20 million shot. So you're looking for something that's 1 in 20 million in something that's already pretty small. So it doesn't work, extremely well. It works at late stage, it doesn't work early stage. And the difference with what we're doing is that we can look at all of the nucleosomes, because the epigenetic changes that occur in the nucleosome occur right across all of the chromosomes. They don't occur to one particular point. And the reason that happens is because they're enzyme based, and the enzyme doesn't look for a tumor associated gene, it just does the whole thing. And the course of evolution of the cancer does the rest. So our aim, and the project that we're doing, is to look for those cancerous, is to look for the nucleosomes themselves, which are elevated in cancer anyway, and also for the particular associated cancer changes. And here's a cartoon of a nucleosome, really just to show some of the potential changes that can occur. So each of these points that's marked here is a potential place where you can get a signal, an epigenetic signal. It's a chemical signal. And in many cases, there are multiple changes, multiple different signals that you can get. And the 4 proteins listed there, H2A, H2B, H3, and H4, they also have multiple isoforms. So for many of, there are 300 or 400 at least human different potential proteins that you can have there, and all of those are epigenetic signals as well. So there's a lot of things that we can measure and that we are measuring. And what we've done recently, I think probably you will have heard, and I've already been asked a couple of times today, we've recently revisited how we do this, and actually the platform is now really superb, and we can detect individual signals. We can prove that it's quantitative. We can prove that we detect only that signal, and not, if that signal's absent, we can prove that we don't detect anything. And if there's a similar signal right next door, we can prove that we don't detect that. So the platform is absolutely superb now, completely reproducible and ready for product. So with that new platform, I'm very happy, by the way, to talk about the technical details of that. What's different about the platform now? Why is it so good? Probably not, maybe not, this is not the right place, but if anyone wants to ask me during drinks afterwards, I'm happy to go into that in detail. So we've now put some of these assays through some clinical studies, small numbers at first, but we will be moving it into the big numbers. And here's just some examples of the sorts of results that we've got. So in a lung cancer cohort, it's small numbers, it's 76 people, but with just one of our assays, we were able to get an AUC of 85%. And that was cancer versus healthy, but it also discriminated lung cancer versus other lung diseases. And then we looked in a second lung cancer cohort, that was 152 people, and did exactly the same thing and got a very similar results, about 80% AUC. So the The platform itself is analytically reproducible in scientific way, and the clinical results are excellent and reproducible. So we will be putting that assay together with others to increase the AUC further. In a separate experiment, we looked at colorectal cancer. That was 123 people, and with the same assay, it detected it had an AUC of 72%. But when we made a 2 panel, added one more to it, that went up to 84. And I've got a picture of what that looked like. So the green subjects on the left are people that were sent for colonoscopy, but actually they had a clean bowel, there was nothing in there. And then the 4 red groups are stage 1, 2, 3, and 4 cancer. And you can see very easily that we detect all of the 4s, and we certainly detect most of the 2s and 3s, and we even detect a very large proportion of the stage 1s. And actually detecting stage 1 cancer is something that there isn't any other test out there that does that. The Septin9 test, to the best of my knowledge, detects relatively few stage 1 cancers, and things like CEA don't detect stage 1 at all. And on the right hand side, there's a ROC curve which shows, I don't know if you're familiar with ROC curves, but it shows how the sensitivity of the assay varies if you change the cutoff at which you do it. So these are early results. We will be taking those forward into bigger sets. In the lung cancer example in particular, we're now collecting 1,200 sample set of people that have had low-dose CT scanning. These are high-risk people that were sent for the best current lung test. The good thing about low-dose CT scanning, and really X-rays as well, is that it does find most cancers, because if you've got a lump in your chest and you X-ray your chest, you will find that lump. The bad thing is that it has a 40% false positive rate, because if you essentially you're taking a picture, and if there's a lump or a nodule in someone's chest and you take a picture of it, you can see it and you know where it is, but you have no idea whether it's malignant or not. And so 40% of the positives are false positives. There is something there, but it's not a cancer. And the study that we're collecting at the moment will be to see how well we can discern whether the people that have a nodule in their chest actually do have cancer. That's collecting now. We will get the first results of that early next year.